Hello everyone, welcome to our next video lecture on the topic NGS analysis. So in this video we will be learning how to do a quality check of the the files, fastq files that we have, and also to how to remove bases that are of small quantities and also remove reads that are of small quantities also. So in this lecture we will be using several tools like as fastx toolkit, cutadapt, as well as a fastq pair tool get the work done. So if you go into a previous video, we have known about we have removed the contaminants that are present in the fastq file, and we have generated this file fast clean reads 1 and clean read 2.fastq, right? So if you look into the report, fastqc report of the file, you can see that there are several parameters that we need to look into and correct accordingly. In both the files there are some issues there. So if you look into that, you can see that the per base sequence quality and per tile sequence quality is having some error there, right? So it is showing this red cross and all, while the exclamation mark is shown for yellow exclamation is shown for the per sequence GC content. But I will rule the per sequence GC content, because if there was any issue in the GC content, then it would have been ruled out when we are removing the primer as well as removing the genome contamination. And also, as it has an innate nature of Pseudomonas aeruginosa genome, then we can basically rule that out and focus on clearing these two parameters here and there. So in both the files there are some issues with that. So first what we will do is that we actually need to install some of the tools. As we have discussed, we will install tools like cutadapt. For that I will sudo apt install, type in this particular command, because cutadapt is also directly present within the distribution. So I will just press enter. I will type in my password and let the package be installed, because it is already installed in my system, so I don't need to reinstall it out. I also need to install a tool called as fastq pair. So I will use the command sudo apt install fastq pair. So it is already installed. We have installed both of these tools, and one more we want to install is the fastx toolkit. So but we cannot use it directly the sudo command to install it, because it is not present with the distribution. We need to use bioconda to install it out. So I will just clear this terminal out and I will configure the channels. Conda config channel defaults. I will just prioritize this channel first. Okay, I will also prioritize Conda Forge. Sorry. Okay, and we also want to prioritize Bioconda channel also. So I have prioritized this. Then I will create an environment like conda create, using conda create option, -n fastx environment using Python report and version, that current version that I am having. So I will just wait for some time until it is created. Okay, so I have already created this out, so I will just type in, for in your case you just proceed with creating the environment. Okay, so then in order to install this particular fastx toolkit into this fastx environment, first I need to activate the environment itself, right? Conda activate. So it will activate the environment. You can see the shift from base terminal to the fastx environment here. Then I will type in conda install -c bioconda fastx toolkit. Okay. So what it does is that it actually starts to install this particular bioconda environment into it. So I already have that toolkit installed in this particular environment, so I am not going to proceed with that, and you should basically install this out. So these are the basic things that you need to have. First of all, you need to have the cutadapt, then fastq pair, and fastx toolkit. And using this, we will do a quality control of the fastq files that we have, sort out the parameters that need to be sorted out in this case. So as I have already created it out, this clear it up, and then I will go through the fast files that we have. So I will just open one file at this point of time. I think I will just open this first report. I will start this first report, and then we will move on to clean the second report as well. Yeah, so I think that will be okay. So I will just list out what all files that I have, and also I have kept the fastest environment active here so that I can use a quality filter to rule out the parameters. So first of all, what I do is that I will analyze the report. When I am analyzing the report, I have a problem with the per base sequence quality and per tile sequence quality. So what I will do is that I will try to sort out the per tile sequence quality. So the per tile sequence quality means that within the flow cell there are different cells that are present, right? So each of these fragments represent different cell. Each of this rectangular shape that are seen here represent each of these cells. So there are about 1 to 120 flow cells that are present here, most probably here showing here, or 118 flow cell, whatever it is, no issues in that case. So in that particular range, so if you are looking into a first flow cell, you can see that in the first flow cell most of them are into the cold color, meaning that there is not much error that are present here. But if you take like in the 49th or in between 49 to 52 cell, you can see that there are some hot colors that are present here. You can see some red colors that are present here, meaning that at that particular range, if you look into that particular flow cell 49 to 52, in the when the read is getting generated, whenever a read is getting generated in this particular flow cell, that is 49 to 52, at the position of this read, like second position, third position, at that particular range there are some arrays that are coming up. So it means that if you are considering a read from 1 to 100 base pairs, right, within that read at the second or third position there are some arrays that are happening. And also similarly you can see some yellow colors that are appearing here, which are also hot colors at this particular range in the flow cell. At this particular range itself, you can see that between 72 to 96 in between them you can see some arrays there. So in this case, what we can do is that we can reduce the number of reads that are coming in between having these bases, because you can see that some arrays are coming up in the middle of the base, in the middle of the reads, and some are coming at the end of the reads. So if they are coming at the end of the reads, basically you can trim them out. But if they are coming in the middle of the reads, then we need to actually do a quality filter to remove the reads that are having a particular parameter that is getting affected there, right, having low quality and all. So what I will do is that I will use the fastq quality filter that is present within fastx toolkit. So in order to call out that particular quality filter, then I will type a fastq quality filter. Okay. Then there is an option called as q. So the what q does is that it marks a base pair base that is present within a read based on the information we provide. So as you know that in the fastqc file, right, fastq format, the quality is basically provided there. So whenever the quality is provided there, it is actually denoted in a mark q format. So there are qualities that are coming up around 10%, 0%, 30%, 40%. So most of the qualities that are below 20% is considered, or 15% is considered to be problematic. We need to remove it out. So what I will do is that I will give the least quality, so that fastq filter actually, what a fastq filter does is that it goes through the each read pair that is present within the fastqc file, and if the read pair is having a quality equal to or less than 15, it actually flags those reads out. So suppose that there is a read of 100 base pair, there are about 20 bases that are present within that read having a quality less than 15 or equal to 15. It is getting marked by this fastq quality filter there. It first of all marks it out, and then in order to remove those reads, what we have to do is that we have to give out another parameter called as -p, or a command called as -p, and then we have to give out a value. Basically, if you look into Q, the value actually ranges from 15 to 30, because based on the stringency of the removal of reads. If you are increasing the value, meaning that you are increasing the stringency, so more reads will be lost out there. If you are increasing the value of P, basically if the value of P actually ranges from 65 to 90. So if you are increasing the stringency above 90, then more reads will be filtered out there. So whenever you are increasing the value of Q and P, the number of reads that are getting removed will also get increased. So that will affect the assembly also. You can remove more reads that are unreliable, but if you are doing an assembly and everything, or a further analysis, I suggest that you give a proper threshold value, might be a less value of Q and P. Start from that and look into a fastq report, and then proceed with what all values that you can use. But I suggest that you use a less value of Q and P here, because as it increases, the number of reads get also discarded, and it will affect this assembly, because the assembly actually depends on the number of reads rather than the quality of the bases. So I will keep it in this value. So what happens is that the Q parameter actually flags the quality that is present within the reads, quality of base that is present with the reads. If a particular base within the read has a quality less than 15 or equal to 15, it marks those bases. And what P does is that if the read consists of about 90% higher of this particular mark quality reads, if the read consists of about if 90% of the reads consist of about bases that are higher than or equal to 15, then it will keep that particular FASTA file. In this case, if the read have a quality less than 15, then it will remove it out. So it basically gives you an idea like that. So fastq filter, fast quality filter -q 15, I have put -p 90. So that 90% of base should consist of 90% of those reads should consist of a bases that are above 15% mark quality. So if it is less than that, then that particular read will be discarded out. So this is in base quality level, this is in read quality level. So you have to understand the difference there. And afterwards I will give in the input file, that is -I. I will just copy this out, I will just paste it out, and I will also give an output name. So basically this is the input. It goes through input, and it flags this particular parameter within the input, remove the reads accordingly to it, and gives a output file based on the name that you are giving. So this is the first quality first or forward read file, right? So I will just give in the name 1, and I will type in the parameters, because I like to give the parameter name, because I would know like what all changes I have done in this case. If I had to change that particular parameter again from the initial parameter that I am having, from the file name itself, I would understand. So I basically put name according to this, like first of all I started with the quality trim fastq quality filter. I use this command q15 and p19 within the fastq file. So I have that particular file, initial file already, right? So I will get an idea like that. Then I will press enter. So it will basically take some time. So basically here the analysis is done. You can see that the fastq file is getting generated there. I will just clear it out, and then I can list it out. You can see that the first file has been generated. So I will use fastqc to generate the HTML file of it. Okay, so HTML file is generated. So I will just type in explorer.xp. So it generates a particular report there. So if you can see that clearly note out the difference there. Okay. As you can see, many of the hot colors that are present here are removed in this case. So several of the bases that are present that were showing such error which have low quality range were at this range where the sequence length is much higher, right? That's because of the prosody gets low whenever the sequence length actually or the insert size actually increases. That's why most of the low quality bases are shown at the end of the or the farther end of the insert size on the longer insert size. So whenever they are removed, you can see that per tile sequence quality is also getting much better here. But we also need to do some. We are actually getting to the target, but we also need to do some more trimming and everything because we need to solve these parameters out, right? So what I will do is that next I will use a tool called as cutadapt, because we have cleared much of the problematic bases that are present here, but here you can see that there is some problematic bases that are present here, and especially at the starting end of each of the insert there. So we need to remove that out. So what I will do is that I will use a tool called as cutadapt. Okay. So basically this tool enables us to cut the bases from the end. So either from the starting end or at the last end of the insert. Basically it depends on the command that you are using. So basically there are 3 different commands that we probably use in case of cutadapt. So if I here in this case in the file I need to remove from the starting end, right? So I will use a command called as -u, and I will just go through the file to get to know how much bases I have to remove. So if you look into this file, most of the bases are present till here up to I think 12 to 13 to 18 to 19. So I will use cut till the first 15 bases of each of the insert rate. So I will just give a command -u, this one, right? So then I will just type in my input file. Okay. So I have just give the input file, and I also need to give the output file. So okay, so I will just give the output file, and I will add an extension u15 because I know that I have used the cutadapt tool, u command within the cutadapt tool, and trim the first 15 bases within each read, right? So I am dot fastq. So from this file I have generated this file. So based on the addition of commands that are present within each file, you can understand basically, okay, these are the commands that I use, and this is the input file that I used, and from this input file I have generated this output file. So that gives you an flexibility to know what you have been done. So that's why I prefer to use this particular naming of the file because it is quite understandable from this in this case. So if you are using it, you can use it on your own, no issue in this case, but based on your comfortability you can name the files, no issue at all. So I will just use press enter, so it will take some time. Yeah, so it also generates a report. You can see that how many base pair was processed, this much base pair processed, and a total return file, how much like what is it from this, how much base pair was present initially, and how much base pair is present now. 85% of the sequences or base pairs are preserved in this case. So that is produced, right? So I will just convert into a readable format like using fastq. I will just copy paste it out. Okay. I will just clear this out. Okay, I will just exit out and then use explorer.execute. I will copy this file name. Okay. So as you can see, most of the base pairs are getting trimmed here, right? So there isn't much change in the file, and also if you can see, there are some bases that are having some problem here, and especially at this particular end. So you need to clear that out also. So I am suggesting that 78 to 79, 79 to 84. So you need to trim the ends of the extreme ends of the insert size also, but you can see that the per tile sequence quality is getting much better. It is similar to per sequence GC content. Basically there is no issue of keeping the exclamation mark in there. It's just a warning sign. It means that whatever parameters are there for this particular criteria, it is at the threshold. It's not into the extreme end. It's not similar to what a error sign is showing there, right? So you can basically keep that, but I need to clear this out also. So if you are showing in the per sequence base quality, per base sequence quality, you know that you have to trim this out. So basically I think that I can trim it into the 81 length range there. So the sequence length is about 85. I will see whether I can trim it into the 81 length there. So in this case you are generating FTP file. Similarly I will use the cutadapt, cutadapt command that you have used, right, to use the, right. Instead of as I have trimmed from the from the farthest end, I will just type in a command 80 and I will trim each insert base into the 81 length. So the file insert length will be 81 here. So I will just give out the input command. Here the input command was from the U15 one, okay. And I have already also trimmed it into using the L command within the cutadapt tool. I have trimmed it into an insert size of maximum range of about 81 bases there. So I will just press enter. Okay, so it has again trimmed it out. You can see the stat here. I will just type in fastq. I will just copy paste this file out. I will just press enter. Let it do it work. And then I will type in explore.security. Yeah, so you can see that in the final report, you can see that it has been sorted out. There's exclamation mark, leave it out, no issue in this case. And you can see that the per base sequence quality is getting cleared out. In the previous file you can see that it was showing an error. Whenever we cleared it out, you can see that it has been cleared out. So this is a proper report. It's not like it should be in the green line, but if the parameter or criteria is showing a tick mark here, you can proceed with that, no issue. Tick or either exclamation mark. I'm not going to change the per tile sequence quality again, because if I am doing that, then I will lose more reads in this case. And if you can see that the number of reads that are present here are 9,93,192. That's my reads are present here. And for each read, the sequence length is about 81. And there is not much duplication that is present here. That's good. So yeah, so basically this is how you do a quality control as well as a quality trimming of the insert that you are having or a filter that you are having, so that you can start from the initial report that you are having and end up with this particular report where most of the parameters or criteria has been sorted out. So I hope you have understood this. So next of all, afterwards we have to do a for the second file that we are having, right, reverse read file you are having, you have to do a quality check of that also. So I will just do the quality check also and show you the final report that I am having, and then proceed on to do a sequence assembly and all. So basically what I have done is that I went through the second, that is a reverse read file. This is the particular row format that we are having from the fastq analysis and all that we got. So you can see that there are about 90 lakh sequence. Both the reads, forward read and reverse read were having this particular number of total sequences there. So but what I have done is that first I used the fastq quality filter. These are the parameters, the Q15 and P70 that I used. I used the minimum quality control parameter to run the file, because when I was using it into P90, right, most of the sequences were getting lost. The reason why is that because if you look into the per tile sequence quality that I am showing an error, most of the mismatches or mistakes in quality of the reads were coming in between the reads. So whenever that happens is that most of the whenever I use a parameter of Q15 and P90, most of the reads having the particular mismatches in the between will always be labeled out. So the sequence, the number of reads that are present within the output file was less than that of 7 lakh or nearly about 6 lakh sequences were there, reads were there. So that's why I changed the parameters. I changed the value of P from 90 to 70, meaning that I reduced the stringency of the cutting parameter there. So in that case you can see that it was about 90 lakh first, so it ended with 30 lakh, meaning that some of the sequence were read, not much of that. So afterwards you can see that some of the quality parameters were actually like changed there. You can see the shifting graph towards the green area, right, but not that much. And the per tile sequence quality, if you compare with the previous file, you can see that it is getting much more better towards the code colors and all. So what I did afterwards was that I checked into the per byte sequence quality. You can see that it has a sequence length about 100. From the table you can see that there are a sequence length, each read have a sequence length of about 100, and much of the reads that or bases that within each read is falling into a like filtering out area at the range of about more than 80 or 81 base pair in size. So what I did was that I used the cutadapt tool and used the parameter L82. I didn't use the parameter U because there was much problem within the first starting area of the insert, right? The problem with was majority of the problem was within the last area of the or the extreme ends of the read, the final basis of the read, each of the read. So it was quite evident from this particular graph also, where you can see that within the after doing the fastq quality filtering and all, you can see that much of the problem was within this area. So I used a cutadapt to the parameter of L82 to trim out the read from the extreme ends. So you can see that the maximum length came out to be 82, and when I did that, you can see that the per byte sequence quality got also sorted out. So that was also sorted out, but you can see that the per tile sequence quality is again showing an error, but I cannot proceed above this, because if I proceed above this, then the total sequences or total reads that are present in the fastq file will also go into much lesser value, and it will affect the assembly. So I will stop with this. Why? Because the forward and read I have sorted out much of the issue. There isn't much of the issue that I present here. It just like it is at the exclamation mark, but it's good actually. It is at the threshold level, right? So no issue in this case. Even though the reverse read file is having some error, but I have the reliability of the forward and read file there, right? So as I have that particular file properly quality checked and all, I will proceed with the assembly and everything. So I have these two files. I will just go into a terminal. Basically, you know that this is the final file that you are having, the last file, and this is the file that we are having. We will use these two files to do a sequence assembly. So but before that, most of the tools that you are used for sequence assembly in bacteria like BWA and SAM tools, it is suggested that both of the files, that is a forward read file and the reverse read file, should have the same number of reads. It is required for maintaining proper alignment there. So what I will do is that I will use the tool fastq pair. Okay. So I will call out the fastq tool, fastq pair command in this case, and then I will copy these two files, that is the final process file there, right, this one and also this one, and then press enter. So it will go through this particular read and file, and it creates two different files, paired and single end file. So in paired file you can see that in the left paired file, that is the forward read file, and in the right paired file, that is the reverse read file, both of the number of pairs or reads that are present on both the files are the same, and from the leftover reads they have created a different file because they have said that okay, this file these reads are not that needed. We have sorted the both the forward read and reverse read file, and we have generated this paired end file. So this paired end file will be used for assembly. So before that we will just check how the files are looking. I will just clear out the terminal. I will just list out the commands, and I will do a fastq to convert the paired end file. So these are the paired end files that are generated, right? So I will just copy their name, and I will just copy the reverse read file also, paired end file. Okay, so let it convert it into a readable format, and afterwards we will analyze the fastq file that is being generated. So I will just clear it out. I will just list it out. I will just use explorer. Let execute, and I will load the HTML file, and I will again do the same for the nest file, and then so that we can basically compare them out, right? Okay, so basically we have loaded out the file. I will just sorted this out. So from the, if you compare from the first to second file, you can see that there are several changes that are being made. So the total number of sequence present were about 9 lakh. Here total number of sequence that are present were 8,24,570. So the only the number of reads were different in this case, but all the other parameters remain the same. So as the number of reads change, also you can see that the per tile sequence quality and other stuff also change. So that is quite usual when you are doing a fastq pair, and also no issue in this case. If you look into the first file that you have generated and compare with the second file, you can see that there are also this much change that is happening here, but all the other parameters quite remain the same. So it is similar to what we are seeing here. So if you look into this both files, you can see that the number of sequences or the number of reads that are present in both the files are same. This is necessary because some as I have said, some of the tools that you are using, that is BWA, require that the both the forward as well as the revert read file, that is paired end file, should contain the same number of sequences. It is required for proper assembly or proper alignment and all. Yeah, so basically this much we will talk about in this lecture. So you have learned how to do a quality checking of the fastq files that we have using tools like cutadapt, fastq quality filter in the fastx toolkit, and fastq pair for proper assembly and everything. So yeah, I hope you learned several things from watching this video. Yeah, so thank you.